Well, welcome. I'm Susan Forrest, and this is your review for Chapter 5. So we're going to do just a short review here over Chapter 5, and I am just going to cover the basic things. This is not a replacement for the video. If you haven't watched the video lectures, you should go back and then come watch those and then come back. So if you haven't watched the video lectures, bye. But if you have, here we go. So first of all, this chapter is divided into two basic parts: the cell, as the name says, the eukaryotic cells, and the microorganisms. So what kind of eukaryotic microorganisms do we have? Things like fungi and the protozoans and the algae and helminths, which are worms. And so we're going to be going through some of the various different structures of those different microorganisms. Particularly, we'll be looking at the fungi because fungi are a big kind of issue with a lot of disease. They do cause quite a few different diseases, and a lot of people think that they're bacterial infections when they're not, and so they take antibiotics and those don't work because these are eukaryotic organisms, and antibiotics only really work with prokaryotes. So this, you'll notice that we're doing this chapter before we do chapter 4, which is over the bacteria, the prokaryotic cells. But you should understand that this is a cell that you should be familiar with, because if you've had anatomy and physiology, you have already gone through the eukaryotic cell. You've already discussed it. So we start here with something you should know, and then we build on that. And so in the future videos, then you will learn more about the bacteria and you'll learn more about the viruses. So, but this is something you're more familiar with and should be something that's just a review. But the microorganisms are new, and you may not have studied these particular microorganisms, especially if all you've had is anatomy and physiology, if you haven't had any kind of biology class before this. So we're going to go on into those two sections. We're going to start with the cells, structures of the cell, and then we'll get into the different kinds of microorganisms. So in this section, we're looking at the fence, the gate, the main office, the walls, all the different structures that you would find in a factory, different kinds of factories. And so typically there's going to be a fence around it, and sometimes it's a fortified fence and sometimes it's just a chain link fence, so that's similar to the cell membrane. And so the gate is like a channel protein or something that allows things to go through that normally can't make it through the fence. The main office then is going to be like the nucleus, and these are some structures that every year I kind of add a little bit to this analogy, but the walls of the main office would be like the nuclear envelope or nuclear membrane, and the pores within that nuclear envelope would be like the doors in the walls. And so that's how things get into and out of the nucleus. The computers are like the chromosomes. So you have lots of different computers, and usually there's a computer and then there's a backup that holds things. The files on the computers are like the genes that are on the chromosomes. The factory worker, or the worker training center, so you have to know where the worker is. The worker is a ribosome. So the worker training center is like where you make the workers. And so where we make the ribosomes is the nucleolus. And the factory foreman is like an mRNA. So I get very detailed in this, but probably the main ones you're going to get are these things like the fence, the gate, the main office, the assembly line, which is the endoplasmic reticulum with or without workers. And so that's like without workers, it would be the smooth endoplasmic reticulum. With workers, it's going to be the rough endoplasmic reticulum. And this is sort of the endonuclear membrane system, where it goes from the nucleus down to out of the cell. So the process of making product, you would have ribosomes being made, and then you have a factory foreman, which is like the mRNA, that would take the information out to the assembly line, and if there's ribosomes on that assembly line, they would put the pieces of the amino acids together, or put the amino acids together to make the protein. And then the packaging and shipping department would modify it and send it on to a truck or semi-truck, which is like, well, so the packaging and shipping is like the Golgi apparatus, and it would modify the protein and package it up into little vesicles. And those secretory vesicles would be the semi-trucks that would carry the product out of the cell and deliver it to different parts of the body. So that's just kind of a big overview of that. Now there's another slide to this though, that kind of some other structures like the solar panels and the power transformer, warehouse, water tower, cafeteria, steel beams. And so the solar panels are things like your chloroplast. So some eukaryotic cells are photosynthetic, and they have chloroplasts that will take energy in from the sun and convert it into another form of energy. So this is a very good analogy because that's exactly what a solar panel does. A power transformer is like our mitochondria. So you may have heard of it as the power plant of the cell. And yeah, but what it's really doing is it's transforming it to usable amounts of energy. So if it has glucose, it starts with glucose and then it transforms it into ATP, which is a usable form of energy. That's what a power transformer does. It takes the energy from the high voltage power lines and then converts it into a usable amount that comes out of your receptacles in your home. So the warehouse would be like a vacuole and or a vesicle. The water tower would be like a central vacuole maybe in a plant cell. The cafeteria would be like the cytoplasm because it's got all the nutrients and things that are needed. The steel beams and 2x4s would be like your cytoplasm or your cytoskeleton. So the microtubules, actin, myosin, and any intermediate filaments, whatever gives the cell its shape. A lot of times it's the steel beams and 2x4s in animal cells. So that's a little bit of an overview of those, and hopefully that will help you, because there will be a question on here. You do have to know what these structures actually do, but you want to know the analogy as well. And I feel like if you know the analogy, then you know the cell type, or what does the organelle do. So the plastid organelles are the organelles, the mitochondria and the chloroplast. And so their structure is kind of unique. The mitochondria in particular has a lot of folds in it, and the folds give the endoplasmic the folds give it more surface area. So this is where the electron transport chain takes place, and then there's this nucleoid region kind of, it's where the double-stranded DNA that is a single loop of double-stranded DNA, and then it has little ribosomes, and those ribosomes are 70S ribosomes, which have a 50S subunit and a 30S subunit. And that is distinctly different than the ribosomes that we see in the cytoplasm of the eukaryotic cells. And so this is unique and different. But these ribosomes make the proteins that are coded for by this DNA for the electron transport chain, the enzymes needed for Krebs cycle, you know, all the different precursors and things, the enzymes that are needed for that to happen are encoded by this DNA. And basically the endosymbiosis theory says, you know, that a bigger cell that couldn't do aerobic respiration took up the cell that could do aerobic respiration. And in an environment where there's lots of oxygen, this was more beneficial because it could use the oxygen and it was much more efficient at making ATP. And then later on, others came along and engulfed not only this little cell that could make ATP, but also photosynthetic bacteria possibly that could do photosynthesis. And so this is kind of the structure. They have these little folded areas, these not folded, but they don't have the folded inner membrane, but they have these little thylakoid discs they have their own ribosomes, and they have their own nucleic acid as well. And it's the same. It's 70S ribosomes and single loop of DNA that's double-stranded. And so this is for photosynthesis. And the chloroplasts and the mitochondria divide on their own. They use binary fission, which is another structure or characteristic of bacteria. The size is very similar to bacteria as well. They're about 10 times smaller than the cell itself. And so they're a very large organelle within the cell, but again, they're about 10 times smaller. So this is a lot of evidence for the endosymbiosis theory and how the first eukaryotic cells came to be. And that's something you need to be able to talk about. And it's something that I want to make sure that you review before the test. Another characteristic or thing you're going to have to know for the test are these structures. And so this is also on your lab exam, so there's another lab review over this. But I put these in the PowerPoint as well, so you can, and I'll make this PowerPoint available to you. But know all of these different structures because I will probably have this picture again on the lab and the lecture test. So the lab exam 1, which goes with lab 3, and test number 2, which has the structures of the cell. And you need to know what all these structures do and be able to. So this slide is showing the answers to the previous slide. So again, you'll have some questions where I'll have things pointed out and you have to identify the structures. So use this as another tool. If you want more information, you can go to the lab exam, lab 3 video, and it'll go over that. So we're going to go on to the next slide. Okay, so on section 2, we're looking at the eukaryotic microorganisms, the fungi. And so I'm going to draw a few things on here just to give you an idea of what these things look like. But the fungi, the hyphae, is just like a single strand. When you have a mycelium, it's like a whole bunch of strands, little filamentous, and I'm not doing a very good job on this picture here, but you get the idea. So if you just have a single strand, that is a hyphae, and you have a lot of these, like a single blade of grass would be a blade of grass. And if you have a lawn of grass, then that would be comparable to the mycelium. And the blade of grass would be like the hyphae. So just one little filament is a hyphae, and you have these mycelia that come out, and they kind of are filamentous like this, usually coming from one center point and then spreading out from there. So these fungi have asexual and sexual spores, and that is one of the ways that you can differentiate between them. They sometimes are septate and sometimes non-septate, so they have these, if you look at them under the microscope, sometimes they're septate, and so each one they have little cross walls, and they're like each cell is an individual cell. But the non-septate are more like, there's lots of nuclei in here, but there's no cross walls, there's no divisions between them. So how we classify them is by their sexual spores. And so that's one of the, or how we group them. And that's pretty much the main things that you need to know for the fungi. One other issue is that when they're in the yeast cell, this is a single cell stage, and so unicellular. But when they're in the hyphae, they're filamentous. I don't know that I would call them multicellular. They're kind of somewhere in between a unicellular and a multicellular. So filamentous is more like the term I would use. But when they're, and they also call this the mold stage. So it's like filamentous or they're single celled. So they're called dimorphic because they have more than one form. So form and di means two, so they have two forms, the yeast and the single cell, and which is single cell. And they have the, it's more hostile, it's hotter, less nutrients. And then when they're in the mold form, that's more of the good environment. And so it's cool and moist, and it has a lot of, I ran out of room there, but you get the idea. So it's cool and moist, and it's got lots of nutrients. Hostile usually is a hotter, and if you think about it, if you're going to make yeast rise in bread, you're going to warm it up. And so that's kind of the main thing. This is pretty much it for what you need to know about the fungi, and this is more than what you have to know for the other microorganisms. So if you think this is going over a lot, it is because this is a little bit more detailed than what you have to know for the others, but that pretty much covers the main points. The algae and the protozoans and the worms, mainly, you just have to know a little bit about the structure and how is it classified. So for algae, a lot of times we think of these as being green, but they have lots of different colors. So it's the color of their pigment is how we classify them. They are photosynthetic, and so they are kind of the basis of the food chain, and so producers. And the different pigments that they contain, they don't just have chlorophyll, some have other pigments as well to collect sunlight and to do photosynthesis. And so the color of their pigment, the primary color of the organism, is the way that we kind of look at these. So they're photosynthetic, they are not multicellular, they're colonial, they're unicellular, and sometimes filamentous. So in our lab, we may have seen some Volvox that's like got spheres within spheres, and they're green. We may have seen some Spirogyra with the spiral kind of chlorophyll or chloroplasts in there. And so that's more like a filamentous, and then this would be more of like a colonial. And then you can have just regular single algae, unicellular. So they can be all of these different kind of types. And most of them are producers, and they're more plant-like, so they often do not have any kind of locomotion. On the other hand, your protozoans are going to be more animal-like, and they're probably not producers, but there are some that are called mixotrophs. So they have locomotion, and they typically are heterotrophs. And so they, it's kind of going off the screen here, but they're not photosynthetic. And so that's like they're a consumer in the ecological system and not so much of a producer. But so where the algae are more plant-like, the protozoans are more animal-like, and they're classified by their method of movement. So sometimes they have little cilia around them. So those are really only found in the protozoa. So cilia is kind of unique to protozoans. Sometimes they'll have a filament like this, a flagella, to help them move, and it's a different kind of movement, but they have two different ways of moving compared to bacteria that only have a flagella. But protozoans are uniquely going to have cilia, so you don't see the cilia in other types of organisms. You don't see them in bacteria. Now you do see them in animals, I will say that. Again, these are animal-like organisms, but as far as like algae, you don't really see the cilia in algae, but you see cilia definitely in protozoans. So you're going to have these cells, they all have a nucleus, of course, and then they're going to have different kinds of locomotion. Some are these little pseudopods, which are fake feet. So like this, and that's how they move around. So they kind of stream in one direction or another. So that's a little bit about the protozoans and algae. Okay, so this is another group of organisms, and this is the last group of eukaryotic microbes. These are not really microscopic, but they do get transmitted by eggs, and that's why we talk about this. So the segmented are the class Cestoda and the class Trematoda. And the flatworms themselves are called phylum Platyhelminthes. And so platies are, flatty is the way I remember this. So I don't do a lot with classification, but I do think you need to know that helminth means worm, and that these are animals. These are animals, they're from the animal kingdom. They are multicellular, and that there's different classes. So you have some, these are the tapeworms, and the trematodes are the flukes. And the flukes we did look at, and the tapeworms we did look at. The tapeworms have kind of a little head like this, and then it has little segments called proglottids, and that head area is called the scolex. The flukes are going to be kind of like a leaf shape to them, and again, I'm not doing a great job of that drawing, but it is leaf, like tremble like leaves, trematoda. So you can remember that they are a leaf shape like this, that is the trematodes, the flukes. And the roundworms, they're just non-segmented roundworms, and sometimes they'll form little spiral kind of structures within muscle cells. There's lots of different roundworms that cause disease. In fact, largely, if you're talking about a worm that causes disease, it's probably a roundworm. Sometimes they're little, sometimes they're bigger, but again, it's the egg is transmitted, and they are transmitted through like food and water, and so that's why this is important. So these are a different phylum. They are Nemahelminthes, or nematodes. But just remember, again, Helminthes means worms. And that's pretty much it. So hopefully this has been a helpful review for you. Just a short little 25-minute video. And take all the time you need to review these and go back to the different slides if you need to. And have an excellent time learning all about the eukaryotic microorganisms.